Hello and welcome to Nikolai's genetics lessons, and today's problem is the following cross is performed in fruit flies. So here's one parent, here's another parent to give F1 progeny of the following genotypes, and here's a genotype of the progeny. This F1 are crossed with another testers of the following genotype, giving the following cross. And here's the cross of the genotypes. What is the map distance between A and B genes? And here is a table what we got as a result of such a cross. For many students, this type of problems is a headache because, first of all, what all these pluses stand for? Actually, this stands for the wild type. We also can say that this is dominant allele. If you like, you can change these pluses to A dominant, B, and C. So capital A, capital B, capital C. Capital A, capital B, capital C. Here is example of this genotype. Take a look. So one chromosome, another chromosome. And this is same chromosome. One animal inherited from the mother side, another from the father side. And according to our problem, this fruit fly has three dominant alleles here. So, or wild type allele. One allele, second, and third. One, second, and third. And these alleles are identical. And we cross this genotype with another genotype. Take a look. Another parent, so this is going to be parent 1. And of course, parent 1 is diploid, so has two homologous chromosomes for each chromosome pair. This is not the only two chromosomes, of course, but we just interested in these two chromosomes. And we cross with another parent, parent 2, whose genotypes is as follows. So one homologous chromosome, another homologous chromosome, and this is going to be the same chromosome. Let's say that this is going to be chromosome number 1, and this is also going to be chromosome number 1 in parent 2. And parent 2 has here allele A, B, and C, and also A, B, and C. So for all three allelic pairs, this parent 2 has the same alleles on the chromosomes inherited both from the mother side and father side. Some people think it's confusing, so you actually can change these pluses to dominant alleles, because plus means that this is wild type, and wild type is dominant. So let's say you can change it to capital A, capital B, and capital C. Capital A, capital B, and capital C. So now maybe it would be easier for you to understand that we just have two parents, one that has two homologous chromosomes. This parent is wild type, so has dominant alleles in each locus. And parent 2 has recessive alleles in each locus. So this is this easy. So this is parent 1 and this is parent 2. And of course, what we can get as a result of such a cross. Even if crossing over would happen in any of these places, we're still going to get a chromosome which would have dominant allele A, dominant B, and dominant C. Take a look. Let's say crossover happen here. We still will get one chromosome, capital A, capital B, capital C, and another capital A, capital B, capital C. So if crossing over would happen here, it's not going to produce new variants. And also the same is true for the parent 2. If, for example, crossing over would happen here, are we going to get new variants of the chromosomes? No. Small a, small b, small c, small a, small b, small c. So it doesn't matter where crossing over is going to happen, it's going to result in the same set of alleles. Now take a look. As a result of crossing of these two parents, so each parent would produce gametes where we'll find only one chromosome, whether this chromosome, whether this chromosome, or recombinant of these two chromosomes. But again, because parents are homozygous in all three loci, even recombinants is going to be identical to these two original chromosomes. So we can say that parent 1 only can give to the following generation only one type of the set of alleles on the chromosome number 1. This is going to be capital A, capital B, and capital C. And parent 2 only can give one variant, which is going to be small a, small b, and small c. So as you see, neither this parent nor this parent can produce any other variant. So we know for sure that in the next generation, which is going to be F1 generation, all the progeny are going to be uniform and going to be heterozygous at each locus. Again, do you remember that we also can change capital A to plus, capital B to plus, and capital C to plus. Basically, capital C and plus just means wild type allele. So if we have here small c, that means recessive. And this parent for each locus has dominant allele or wild type allele. So now I hope this pluses is not going to cause a confusion. Now let's read our problem again. So when we got F1 generation, take a look. This is exactly what we got in our drawings. This F1 are crossed with another parent who is homozygous for both chromosomes. So this slash means one chromosome here, another homologous chromosome here with these alleles on these chromosomes. And we call such a cross a test cross, giving the following cross. And here is the cross. So let's draw it. And here is our F1 generation. So F1 generation. And we were told that we cross with another parent who is also has two chromosomes. Do you remember? This is chromosome number 1. And this is also chromosome number 1. And because animals are diploid, they have one chromosome inherited from the mother side, another from the father side. And this test cross is performed with a parent who is homozygous recessive for all three loci. So small a here, small b here, and small c here. So homozygous recessive for all three loci. We call such a cross a test cross because now we can find if there going to be a recombination. Again, if parent 2, so this is going to be parent 2, this is going to be parent 1. If parent 2 would have recombination, it doesn't matter where it's going to happen, with what frequency, parent 2 only can produce one type of the chromosomes. And here is a chromosome with only one variant possible, which is going to be small a, small b, and small c. No any other variants. So this diploid organism will produce a gamete. If it is a female parent, would produce egg cell. If it is male parent, would produce sperm with chromosome number 1, which has only one variant of the alleles on the chromosome number 1, which is going to be small a, small b, small c. So would be recessive for all three genes. But parent 1 would produce much more different variants due to crossing over. The first variant would be, what if crossing over is not going to happen? Then we will have two variants. So one would be, now let me change capital A, capital B, and capital C with pluses. So it would be easier for you to understand later how we are going to solve this problem. So the first variant would be plus, plus, plus here. And the second variant would be, if crossing over didn't happen, it's going to be small a, small b, small c. Basically this whole chromosome. Small a, small b, and small c. Now let's imagine that crossing over happened here between gene A and B. Then we are going to have two new recombinant variants. One would be, take a look, capital A, small b, small c, or we also can say plus small b, small c, plus small b and small c. And another variant would be small a plus small a plus. Again, instead of plus, we can put capital B, capital C here. Now, forget about this crossover. So imagine that we don't have crossover here. Now imagine that crossing over happened here between gene B and C. Then two new variants, plus and small c. This is going to be one variant, plus and small c. And another variant would be small a, small b, and plus. So small a, small b, and plus. And the last recombinant variant that may happen, we can get crossing over both in this place between gene A and B and between gene B and C. This would result in two variants. Take a look. The first variant would be plus small b plus small b plus. And another variant would be small a plus and small c. Small a plus and small b. Now one more time, I need your attention here. It is very important. Parent 2 here can produce only one variant, which is here. So this is going to be combinations of these alleles inherited by F2 generation. From parent 2, from F1 generation, F2 generation can get only this variant of the chromosome. But from the parent 1, F2 generation can get one of these variants. And all these variants, let me circle them, is not going to happen with the same frequency. This is also very important to understand. So this chromosome would pair in F2 generation with only one of these variants. But it doesn't mean that we have here 8 variants and frequency of each variant would be the same, like 1/8, 1/8, 1/8, 1/8 for each variant. No, it's not so. Among these variants, this variant, which we call parental variant, and this is what we see here, would prevail, and another which is going to prevail is going to be this variant. We will find them with much more high frequency than these variants. Some of them would be a result of the crossing over between gene A and B, like in this case and in this case. In other variants between gene B and C, like here and here. And some of them would be result of the crossing over between gene AB and BC. And would be recombinant variants. One more conceptual idea to understand would be that if distance between gene A, B, and C would be the same, then we will find all these recombinant variants except where we have double crossing over with the same frequency. So these two variants and these two variants. But usually the distance between genes are not the same. Take a look. For example, let's say this is two homologous chromosomes, and we have here gene A here, and let's say capital B here, and small b here, and gene C is going to be here. Of course, as you see, frequency of the crossing over would be not equal. For example, between gene B and C, it would be much more frequent than between gene A and B, because distance between the genes here is different, and between genes A and C, frequency would be even with much higher frequency than between gene A and B and C. If a probability of the crossing over between gene A and B would be 10%, so would be 0.10, and between gene B and C would be 20%, 0.2, then probability that crossing over would happen here and here would be a product of these two frequencies, so 0.1 times 0.2. This is going to be a probability that crossing over would happen here and somewhere here. It can be this, or any place, but it's going to be a product. So frequency of double cross would be a product of the frequency of the crossing over between gene AB and BC. So we'll find this is going to be the least frequent combination of the genes. So if we say that this is going to be the most frequent chromosomes combinations in F2 generation inherited from the parent 1, then we also can say that this 2 would be the rarest. Because in order to obtain such recombination should happen in two places simultaneously. Now we can return to our problem when you know all this theory, and let's solve this problem. At all these genotypes, now you can say where crossing over happened. We are interested only in crossing over frequency between gene A and B. Take a look. Now you can say that crossing over happened here because we have recombinant plus and BC. And of course, you see all the chromosome inherited from the parent 2 is the same. Do you remember that parent 2 only can produce this type of the chromosome? So we ignore parent 2 here and we only concentrate on left part. So this is going to be one of the recombinants where recombination happened between gene plus and B, or capital A and B. So let's put this frequency here, 50. And this is also going to be another variant where crossing over happened between gene A and B. And the frequency is 46, so plus 46. And here also a recombination happened between gene A and B, but also it happened between gene B and C. But we are not interested in recombination between gene B and C. We're only interested in recombination between A and B. But still, this is the same frequency. So this is going to be 13. So plus 13. And here's another variant where recombination happened between gene A and B. Do you remember this is double crossing over? So 11 we have to add here, plus 11. And we have to divide by the frequency of all genotypes that we can see in F2 generation. So we have to add all these numbers and we are going to get 1000. I know this because I already solved this problem, but when you will solve this problem on your own with different numbers, you of course have to add all these numbers. It not necessarily have to be 1000, it can be any number. If we add all these numbers in the numerator, we are going to get 120 divided by 1000 is going to be 12 map units. We also can say 12 centimorgans because 1% of the recombination frequency equal to 1 map unit and to 1 centimorgan. One more time, we have found that frequency of the recombination between gene A and B happens with 12% frequency, which we can convert into the distance between gene A and B into 12 map units or 12 centimorgans. But you have to understand that this distance is relative distance. The same 12 centimorgans on different chromosomes between different genes can be significant different number. This only gives us position of the genes relative to each other, but it doesn't tell us exact distance between two genes. Those give us idea of the rough estimation of the distance between genes. So the most important information that crossing over experiments give us is just mapping of the chromosome and tell us the order of the genes on the chromosome. And this is all for today. Thank you for your attention. Please subscribe for my new videos that I post almost every day, and see you in the next video. Goodbye.